Hello, I'm Wakako Yoshioka. Thank you for giving me opportunity to introduce about my project. Today, I'd like to talk about activation of endogenous mutated GNE product by small compound. Now, I am talking from Tokyo, Japan. I am studying GNE myopathy at National Center of Neurology and Psychiatry under Dr. Nishino and Dr. Noguchi. And this red circled place is our center, and Department of Neuromuscular Research is here in the red mark. And in spring, we can see cherry blossom, and in fall, autumn color of leaves, and they are so beautiful. So please visit us when you come to Japan. So these are the key points. Activation of GNE would enable continuous sialic acid supply, and we believe that it would be very effective to treat GNE myopathy. So today, I would like to introduce our brand new approach to find small molecules which activate GNE enzyme. And this project is funded by NDF. As you know, GNE myopathy is an adult onset distal myopathy caused by Sialic variants in the GNE gene. The first appearance of symptoms typically occurs in around the third decade of life, and distal muscles are preferentially affected and develop to involve other muscles, leading loss of ambulation. And as you know, biallelic mutations in GNE gene would lead to hypociliation, and this is known to be the cause of muscle weakness and muscle atrophy in this disease. So let's take a close look at the pathomechanism. There are two pathways in sialic acid biosynthesis, and one is the newly producing pathway called de novo pathway. And this is a cell, and in this cell cytosol, cells use UDP glucanate from glucose and make sialic acid using enzyme reactions. And the other is salvage pathway, and this is the pathway to take up sialic acid from foods or drugs. So GNE gene is here, and encoding the two key enzymes of sialic acid de novo pathway. And if there are biallelic mutations in this GNE gene, it would lead to low enzyme activity and then result in hypocilation. So the decrease of sialic acid production is the essential cause of the muscle weakness in GNE myopathy. And sialic acid supplementation therapy was conducted. In this figure, there are many forms of sialic acid used in animal experiments or clinical trials. And NeuAc is sialic acid itself, and NeuAc and silyl lactose would be taken up using this salvage pathway, and MONAC, which is now used in the clinical trials, are using this de novo pathway to make sialic acid. And proof of concept has been acquired using model mice studies. So when you treat asymptomatic model mice with oral sialic acid, skeletal muscle atrophy and muscle weakness have been totally prevented. And if you treat symptomatic mice with oral sialic acid, skeletal muscle atrophy and weakness improved. So you can say from these results that the treatment of sialic acid supplementation was very effective in model mice. But one of the major concerns to develop extrinsic sialic acid supplementation therapy for humans was rapid metabolization of sialic acid. So these are data of serum sialic acid concentration in 3 patients. Upper graph showed serum sialic acid concentration after a single administration of sialic acid itself, and each color represents each 3 patients. You can see that there is no obvious increase in the sialic acid concentration after a single administration. And the figure below shows that sialic acid concentration after 3-time administration of sialic acid itself for 5 days. Also, you can see that serum sialic acid concentration did not elevate in all 3 patients. I did not show in this figure, but urine sialic acid concentration elevated immediately after administration, so sialic acid is metabolized in a few minutes. But in mice experiments, serum sialic acid concentration was elevated. And this is because sialic acid was dilated in water, and mice were drinking it very frequently through all day long. So this enabled continuous sialic acid supplementation. And I think this was one of the key points of high efficacy in mice. But as you know, human cannot take sialic acid so frequently all day long. So, extended release tablet form of sialic acid was developed for clinical trial to make sialic acid last longer in patient's body. This figure shows serum sialic acid concentration after oral supplementation of sialic acid extended release tablet. You can see in the upper figure that serum sialic acid concentration increased in all 3 patients immediately after administration. And also you can see in the figure below that sialic acid concentration elevated in 3 times administration for 7 days. Even though there is a big individual difference, extended release tablets form could elevate serum sialic acid concentration. And these are the results of human clinical trials conducted using sialic acid extended release tablets. Serum sialic acid concentration elevated in all 3 studies, higher in phase 2 trial in US, Europe, and in phase 2-3 trial in Japan, compared to phase 3 trial in US, Europe. And regarding to the efficacy, improvement of upper extremities was shown in phase 2 trial, but not in phase 3 trial of US, Europe. So regrettably, Ultragenyx had to terminate the development of sialic acid extended release tablet for GNE myopathy. In Japanese study, the results are yet still under analysis, but it was similar to the study results of the phase 2 trial in US, Europe, and they are still working to obtain drug approval. But anyway, you can see that supplementation of sialic acid extended release tablets could be effective, but efficacy was not enough to improve symptoms as in model mice. So let's see the figure of the change in serum sialic acid concentration using sialic acid extended release tablets again. So extended release tablet form elevated serum sialic acid concentration very well, but if you carefully see that at the same time it was decreased in a few hours. So we think if we could provide more stable and continuous supply, it might be more effective to treat this disease. And how to achieve more stable and continuous supplementation of sialic acid? We think the best way is to activate GNE enzyme itself. So like in the right figure. And we think this is the most physiological way, and this would enable continuous supply of sialic acid for a very long time. And then how should we activate GNE MANAC kinase? So traditional new drug development by pharmaceutical companies is one way. But it is not realistic in the field of orphan drugs, and it takes long time for the development of new drug and costs so much. And as the safety is not established, it is so risky. On contrary, drug repositioning is economical and safety is established and is faster for clinical use. So I decided to select drug repositioning to look for known drug that could be a very effective therapeutic option for GNE myopathy patients. But as you know, identification of the drug by drug repositioning is very difficult, especially in the field of finding activator. And the method most commonly used is called high throughput screening. And it is a method to read out luciferase or fluorescence of entire wells of cells and read out by a plate reader. And we have previously performed this kind of screening using lectin staining and use 3 kinds of libraries, which include 400 to 700 drugs. But keeping cell proliferation constant was very difficult, and cell density affected fluorescence. So it was difficult to achieve reproducibility, and we had to explore the other method. We decided to use in-silico screening. We are now collaborating with Dr. Yamamoto and Dr. Sekijima at Tokyo Institute of Technology. They have supercomputer Tsubame. This is one of the world's top large-scale supercomputer. So we asked them to collaborate with us and to perform structure-based screening. In this screening, we use the predicted structure of enzyme and drug compounds. We computationally match them, which is known as docking simulation. There are many clefts in the enzyme, so what we first have to do is to identify which cleft to focus on as the binding target. Then the computer will calculate how strong these kind of compounds or each compounds will fit this target and give us the data of that. And this supercomputer is able to dock the structure known compounds and calculate the binding score very, very fast. So it enabled to identify candidate compounds from 8,042 structurally known compounds in drug bank. Using this technology, the hit rate is known to be about 20 times higher than the previous high throughput screening. So first, we constructed the 3D structure of full-length GNE molecule. And the left figure shows the 3D structure we constructed. But it is a little bit complicated, so I made simple figure on the right side. And it revealed that GNE molecule forms a stable dimer. And dimer means that 2 subunits are joined. And light blue is one subunit, and light green is the other subunit. And they are crossed like X. And this structure seems to be very important for enzyme reaction. And as you know, the protein GNE gene coding have 2 enzymatic activities. One is UDP glucnaq2 epimerase, the lower part of the enzyme in this figure. And the other is MANAC kinase activity, the upper part of the enzyme in this figure. And there are so many clefts in these enzymes. So where should we target? And this is the most key point and the most difficult point of this study. And we have tried 2 approaches: inhibition of CMP-NeuAc binding and stabilization of oligomerization. First, I will explain the first approach we took. This is a simple scheme of the dimer of GNE enzyme. For producing MANAC, first GNE enzyme will be open and UDP glucnaq will bind to GNE enzyme. Then GNE enzyme would be closed for enzyme reaction. And in this phase, UDP glucnaq will be changed to MANAC. And after the enzyme reaction, GNE enzyme would reopen to release MANAC. And this kind of open-close formation change is always repeated to produce MANAC. But when CMP-NeuAc, the final product of sialic acid biosynthesis, binds to GNE, it would act like a glue and interrupt the formation change of open close to stop the production of MANAC. So this mechanism exists for negative feedback, a process to maintain homeostasis by reducing an excessive production of sialic acid. We first try to inhibit the binding of CMP-NeuAc using small compounds. We found a cavity in the back of the CMP-NeuAc binding site, and we thought that small compounds that fit this cavity have a possibility to inhibit the binding of CMP-NeuAc and inhibit the interruption of formation change by CMP-NeuAc. So I asked Dr. Sekijima's lab to perform in-silico drug screening to this cavity and to select compounds which well fit this cavity by not interrupting the formation change of GNE. We selected compounds that strongly binds to this cavity, and we have used 8,042 compounds for screening, and in the right figure, each dot represents each compound. And we selected the compounds that have very strong docking score. Next, we had to measure whether enzyme activity would be increased or decreased using these compounds. And previously, GNE MANAC kinase enzyme activity was assessed by measuring radioactive substrates, though it is considered to be harmful nowadays. So we established a method using ABE labeling. And this figure shows the method I used. First, I prepared wild-type GNE, WT means wild-type and normal GNE, and also mutated GNE. And this time we used the mutation D207V. And we used the cells and that would overexpress these GNE. Then we incubated cell crude with UDP glucnac and selected compounds, and then measured ABEE labeled MANAC. And please see the figure in the right side, the right below figure. And using this method, MANAC production was significantly low in mutated GNE compared to wild-type normal GNE. So it was about 30% of wild-type GNE. So the primary goal of our assay was to find compounds that increase mutant GNE enzyme activity. And these are the results. Please look at the left figure. Vertical axis is the amount of MANAC produced in the tube after incubation. And if it is high, it means that GNE enzyme activity is very high. And WT means wild type, normal GNE, and the other bars represents MANAC production of mutant GNE, which have mutations in D207V. You can see that MANAC production was lower in mutant GNE. And I added selected compounds to this mutant GNE, but MANAC production did not increase. And also, please see at the right figure, I added CMP-NeuAc in the incubation of wild-type GNE and then the small compound. So you can see if you add CMP-NeuAc, the production of MANAC decrease. And I expected if it would increase after adding the compounds, but regrettably we could not find any compounds that increase the MANAC production. So no compound activated mutant GNE nor worked as competitor of CMP-NeuAc. So we tried the other approach. GNE is known to be active after oligomerization. And oligomer is a molecule that consists of a few repeating units, And oligomerization allows protein to form large structures without increasing genome size and provides stability, which would be crucial for enzyme activity. And it is known that oligomerization is required in GNE enzyme activity, and in healthy people, majority of the GNE protein forms the highest oligomer. But in GNE myopathy patients with mutant, especially in the patients with mutants involved in oligomerization interaction surface, enzyme activity would be low by the failure of oligomerization. And this left figure is the immunoreactive patterns of cross-linked products. The horizontal axis represents the size of the protein. So when the GNE protein forms the highest oligomer, the peak of the mountain would be here at 400 kilodalton. And from this analysis, you could see that there are so many mutants that have failure in oligomerization, like these mutants which have peaks in 200 kilodalton, or these mutants which have peaks in 100 kilodalton. So especially in these mutants, stabilization of oligomerization have potential to elevate enzyme activity. And this is just a possibility, but also we believe that even for the mutants without oligomerization failure, like these, we think that acceleration of oligomerization by small compounds have potential to increase enzyme activity. I will show a scheme of enzyme activity and oligomerization. So UDP glucagon would turn to MANAC in the epimerase domain. Then it would be passed to MANAC kinase domain, and MANAC would turn to be MANAC 6P in the MANAC kinase domain. And this MANAC 6P would become sialic acid in the following other enzyme activities of de novo pathway. And we will call it intra-molecular transfer pathway. And also there is another pathway. After production of MANAC, MANAC would be passed to MANAC kinase of the other subunit and become MANAC 6P in MANAC kinase. But if there is an oligomerization failure like this figure, MANAC will be produced in epimerase domain but could not be passed to kinase domain of the same subunit or to the other subunit. So this structure could not produce MANAC 6P effectively because of the oligomerization failure and leading to low production of sialic acid. So what we want to do is to fix this oligomerization failure, like this animation, like this, by small compounds. And we found that in this pink area, this is a very important interaction surface of the two subunits. And we found hydrogen bonds that are connecting the two subunits together strictly and making dimer in wild-type GNE. So we selected the cavity here as the binding target and trying to find small compounds binding this target to fix oligomerization. And these are the results. So vertical axis is the MANAC production in the tube, which represents enzyme activity. And wild type, WP, black bar represents normal GNE, and D207V is mutant GNE. And D207V is a known mutant, which is very frequent in Asian and which is known to have oligomerization failure. And MANAC production of D207V mutant is about 30% of wild type, and we added candidate compounds to this mutant. And as you can see, some of the compounds seem to increase the MANAC production. It did not increase to the level of wild type, but it is said that an enzyme activity of heterozygous GNE, which means that one allele is normal and the other have mutation, are also reduced, and about half of wild type GNE in the lowest. But people with heterozygous mutation are unaffected and healthy. So from that, maybe the increase of the enzyme activity up to wild type might not be necessary. We are continuing to work on this project to find good activator from known compounds. We would repeat the experiments to confirm reproducibility and select the best 3 candidates. And we need to confirm the dose-dependent increase in the enzyme activity. Then we will perform computational screening again for structurally similar compounds to these 3 best candidates, because we want to find the best compounds, the compounds that increase enzyme activity the highest. And also we would like to select compounds with low toxicity for clinical use, and if possible, approved drugs for faster clinical use. And also we will confirm using patient cells with mutant of D207B and other GNE mutants, and measure and confirm the increase in sialic acid production using these compounds. I would like to thank my great teachers, Dr. Nishino, Dr. Noguchi, and Dr. Hayashi, who were always giving me advices, including how to proceed experiments or technical advices, and Ms. Ogawa for her technical support, and all of the lab members, like in this picture, for helping me in many situations. And also, I'd like to thank Dr. Yamamoto and Dr. Sekijima for their exclusive specialized techniques using their supercomputer, and also the platform project for supporting drug discovery and life science research project, because this project matched Dr. Yamamoto and Dr. Sekijima to me. And also, I'd like to thank NDF from bottom of the heart for supporting this study, because we could not do this work without NDF. And these are the key takeaways. So activation of GNE MANAC kinase would enable continuous supply of sialic acid, which can provide better treatment. And in-silico screening, computational screening is a very effective approach to find activator from known compounds. And we found that stabilization of oligomerization by small compounds have potential to be new therapeutic strategy. Thank you very much.